Hello, this is Michael Edwards, CEO at Bioinfo Solutions. This video is part 5 in a series I am doing on crowdsourcing coronavirus data. In the previous section, I examined how the data looked visually and discussed how factors like the animal model, age of the animal, and the strain of the infected virus affected the host lung response. In this section, we will combine all of these data into one signature of host response to infection at day 1 and use it to probe this information for biological insights and therapeutic interventions. Given in this table are the top 5,000 genes whose RNA expression went significantly up or down in the lung tissue at day 1 due to infection in 5 different global gene expression studies comprising 13 different comparisons, each represented in the columns on this spreadsheet. Again, this data is available for free and you can download it at my GitHub account, which you should see a link to. I want to also emphasize that each of these comparisons or biosets used a minimum of 6 study animals per comparison, and altogether these data represents probably hundreds of thousands, if not millions of dollars worth of research data represented in all the numbers we are looking at. Each value on this table represents the scale of magnitude of change each gene went up or down due to infection in the various comparisons. The higher the positive number, the greater its increase in expression relative to the other genes in the same bioset. The more negative the number, the greater its decrease to infected lung. The highest and lowest change in expression is maxed out at 100 and negative 100 respectively. We can compare this information to itself, like determining the patterns that are different in, say, young versus old, or more severe versus mild forms of the virus. Or we can use a wisdom of crowds type analysis to combine all of this information into one response signature that we can then use as our standard for the day 1 after infection. Given in the highlighted column is the average change in gene expression for the 5,000 genes in our meta-analysis. Again, this column is an amalgamation of many different experiments, all looking at the genetic response to SARS CoV infection in animal lung compared to normal uninfected controls at day 1 post-infection. We can now use this information as our early infection signature to identify overrepresented pathways or cellular functions either inhibited or induced by the virus. We can also compare this signature to other viral studies and other diseases to identify similarities and maybe differences that we can exploit. Most importantly, we can look for drugs or compounds that can reverse the types of change in expression that we're seeing due to this infection. We can upload these genes and their associated average gene scores into base-based correlation to probe this signature for biological meaning. Before I get to the biological interpretation, I just want to say that everything that's about to come out of my mouth is all me, and it may not necessarily represent the opinions of my collaborators or of the companies whose software I demonstrate in these videos. I also want to say that no one's paying me to do this, but I get plenty of technical help from the people on my title slide. Thank you very much, Jim, Joe, Enrique, and Mehdi. As I mentioned in my previous section on collecting the data, I decided to use base-based correlation engine because it allowed me not only to identify and download our studies of interest, but I could also use the same program to find overrepresented pathways and biological groups that we can target during early infection. What I really like about this platform is the ability to take our host gene signature for SARS CoV infection at day 1 and compare it to all the other gene signatures in their database, which is thousands. In this way, we can compare what our genes do in related conditions or diseases, or even look for studies using compounds that reverse the changes we are seeing in early infection. But first, we'll look at some of the biological groups elevated or inhibited due to infection. Given on this tab are the biological groups and pathways that were overrepresented in our day 1 signature. The likelihood of obtaining these results by random chance are given by the p-value in the middle, and anything below a probability of 0.05 is usually considered significant. As you can see, most of these groups are way below that standard. Obviously, you see a p-value with a 10 to the negative 131. We are also given a direction of change for the groups based on whether the majority of the genes in this category that were on our signature went up or down compared to our control. And the number of genes in our dataset that followed this direction of change is given on the right. If we take a closer look at the top group, which is no surprise the immune response given the infection, we can get more information on these common genes. On the left, we see our number of signature genes recognized by the platform, which is 4,782. From these genes, we can see that 1,958 genes went up at day 1 of infection, while 2,824 genes went down. The total amount of genes in the immune response category is 460, and approximately 46% of them also showed up in our day 1 signature. The odds of this happening by random chance is given below the Venn diagram. Again, very, very small chance that this would happen by random chance. We also note that 176 of the common genes are going up with infection, which would suggest that this biological system, this immune response, is more active at day 1 of infection. Below these graphs are the immune response genes in our day 1 signature with their associated rankings and magnitude of change. And all of this information is available for download. So we can download any of these lists from any of these categories that we want. If we look down this list, we can see almost all of these pathways that go up are associated with the inflammatory response. And again, we see lots of cytokine interferon signaling along with viral specific responses and genes associated with innate immunity, which again, remember, is the first line of defense against viral invasion. We can filter this list further and look at only established pathways represented in our infection signature. Now, aspects of this inflammatory response pop out, like genes involved in interferon alpha, beta, and even gamma signaling. We also see the activation of genes associated with adaptive immunity at day 1, which again, remember, is our body's second line of defense against viral invasion. I also note that the JAK-STAT pathway is pretty high up on our list, and along with interleukin-6 is believed to be responsible for the cytokine storm that is associated with more severe forms of response to the virus. If we dig further into this pathway, we can see that we had 59 genes associated with this particular pathway at a p-value of approximately 10 to the negative 37th, and 50 of them went up at day 1 of infection. If we look down to see the genes in this pathway, you'll notice that STAT1 and interleukin-6 are pretty high up on our list, ranked 9th and 44th respectively based on the magnitude of their change compared to controls. We also see interferons beta and gamma are also on our list. If I were a clinician and I was looking to inhibit this cytokine storm early in infection that is responsible for so much tissue damage, I would be looking to target these genes. And remember, we can download any of this information and upload it to any other bioinformatics programs we'd like to use. My impressions of the pathways going up with infection are obviously the signs of cells in stress, which is probably no surprise, particularly as it relates to the host response to either a pathogen or viral invasion. We see a huge increase in gene expression due to infection in pathways associated with the immune system or immune response, which includes interferon and interleukin signaling. We also note the induction of pathways that have been associated with bad outcomes in viral response, like the JAK-STAT signaling pathway that we could possibly target with, say, pharmacological intervention. I also want to note that these results are based on the cumulative results from comparisons only looking at day 1 post-infection, and that this list of pathways does change as you move farther out in the infection cycle. In future episodes, we'll discuss how some of these pathways change and some remain the same over the course of the infection. So these are the biogroups that went up, and they're obviously associated with the inflammatory response or response to the virus. Now let's take a look at some of the pathways that went down. I think looking at the genes that go down in an infection can many times be way more informative than seeing what goes up. As we've seen in this example, most of the increased expression of genes with this type of stressor are related to the body trying to alert the immune system that it has a problem with an invader. And as we've seen, this can be more like a scream. But the genes that go down often relate to the systems that the virus needs to suppress or manipulate in order to successfully invade its target cells. If we look at these pathways, we can definitely see some common themes developing among this list. What sticks out specifically are all the biogroups involved in metabolism, specifically those involved with lipids, reactive oxygen species, and toxins. There's also the presence of Alzheimer's disease genes, but this might be related to the biosynthesis of myelin sheets for nerve cells, but I can't prove this without a closer look. We also see a bunch of genes going down associated with cell cycle, which is very different than what we'll see at 7 hours post-infection, where most of these types of genes go up. If we look at the genes in this category, some of the few that go up that are at the top of the list are known inhibitors of the cell cycle progression. I also note that the top genes in this category, cyclin D2, cyclin-dependent kinase inhibitor 1A, or better known as P21, and MYC, have all been shown to be targeted for upregulation by a variety of viruses. Our cell cycle expert on this team, Dr. Enrique Torquia, is taking a closer look at these genes to try to figure out how the virus is using them for infection. There are a lot of interesting things on this list. But for my remaining time, I'm going to take a closer look at the peroxisome category because it seems to connect to a lot of the processes we're seeing that go down with infection. I have to be honest and say that up until this point, I really haven't given much thought about peroxisomes. I always knew they were these tiny membrane-bound organelles, kind of ubiquitous in eukaryotic cells. I also knew they played an important role in the cellular redox state and had important antioxidant enzymes in them like catalase. I was very much surprised with what new functions that have been found out about this amazing piece of cellular machinery. I even read somewhere that it responds to sound. Given on the left is a schematic overview of the molecular machineries and proteins localized at the membranes of peroxisomes in mammals. This is from an excellent review on peroxisomes titled The Peroxisome: An Update on Mysteries 2.0. Peroxisomes contain more than 50 different enzymatic activities. Some of these are listed here, which shape important metabolic and anabolic functions, including fatty acid oxidation and lipid biosynthesis, generation and metabolism of reactive oxygen species, and relevant to our study of the host response to coronavirus, it is involved in activating the innate immune system. Its dysregulation is associated with accelerated aging and other associated disease types that we're going to talk about later as a possible reason for the increased susceptibility to this virus in certain groups of people. But for now, let's focus on the immune response. Given in this diagram is the mitochondrial antiviral signaling protein dependent antiviral signaling pathway, which is a mouthful, which is linked to peroxisomes and mitochondria signaling. We do see an increase of 23 genes due to infection at day 1 associated with this canonical pathway, which you couldn't get by random chance. But there are some key signaling molecules and mediators that are lacking in our metasignature. Just kind of a brief overview of what this pathway does. Upon infection, the viral RNA is released into the cytosol, where it is sensed by retinoic inducible gene 1, or RIG1, and or melanoma differentiation associated gene 5, which is also known as MDA5. These receptors travel to peroxisomes and mitochondria to activate MAVs, inducing a downstream signaling cascade that culminates with the production of type 1 interferons, type 3 interferons, and interferon-stimulating genes, also known as ISGs. In our infection signature, the expression of the viral sensing molecules RIG1 and MDA5 do not show up as either decreasing or increasing compared to uninfected animals, which I find kind of strange. I also noticed that genes associated with the mitochondria and peroxisome organelles, which help mediate the signaling pathway, are going down with infection, like we saw with the peroxisomes, which might be related to a decrease in activity or even number. I also note the intermediate signaling molecule IRF1 is up in our signature. Average ranked for this change is at 64, but the other intermediate, IRF3, does not show up on our list. It should be noted that IRF3 is required for the signaling from both organelles, and IRF1 seems to be specifically activated by peroxisomal MAVs. We do see evidence of transcriptional activation of some of the downstream targets of this pathway, like the interferon genes listed here, with their average change in expression. Although their increase is relatively small compared to the change in gene expression for the other genes in our signature. Remember, our scale is the greatest increase you can have is 100, and the lowest decrease you can have is negative 100. Other investigators have noted an inhibition of type 1 and type 2 signaling in the response to the current SARS-CoV2 virus, and we do see interferons alpha, beta, and gamma on our list, so it's possible that this inhibition happens post-transcriptionally. I also note that one of the receptors for type 1 interferons, interferon alpha receptor 1, demonstrated a large increase in expression in all three of the mild cases of a coronavirus in monkey, but demonstrated no change or actually a decrease in expression in the more severe mouse models used in our metasignature. It's becoming more obvious on the importance of this pathway for viral response. So important that many viruses seek to target this system directly. This next slide shows some of the ways viruses use to target the peroxisome and its associated functions. The types of viruses that target this organelle are many and diverse, so I'm sure it's very important in this type of response. I do point out that the pestiviruses and HIV, which isn't listed here, have been shown to degrade the signaling molecule IRF3 upon infection. And remember, we were just talking about that in relationship to the peroxisome innate immune signaling. And I want to remind you that although we did see an increase in expression of IRF1 in our signature, we did not measure any change in expression of IRF levels compared to uninfected. I also note that some viruses bind directly to receptors on the peroxisome, like the rotavirus class, or produce proteins that bind directly to products from the peroxisome, like the HIV and influenza strains. There is also evidence that some viruses will hijack the entire peroxisome and use its membrane for viral replication, kind of like a crab would finding a shell for its home. It's clear to me that peroxisomes and viruses target each other for modification and/or destruction, but how does this play out in the human population? As I mentioned before, peroxisomes play a key role in aging, or should I say that impairment of these organelles over time is a hallmark of normal aging. As the cell ages, its peroxisomes lose function over time to the point where they have difficulty importing essential matrix proteins. There is also a well-known age-related decrease in activity of the peroxisomal enzyme catalase, which is an antioxidant, which results in the production of more cellular reactive oxygen species. There has also been shown to be an increase in the number of these organelles in some tissues with aging, probably because they need more of them to get the same effect. A similar degradation of function occurs with mitochondria with age, which eventually leads to improper signaling between these two organelles that can impair their normal functions, which I'm sure includes their role in the immune response. Given the preference for the current SARS-CoV2 virus to infect older individuals, I wonder if an impaired peroxisome mitochondria function is making them more vulnerable. Another human condition that has been associated with defective or decreased peroxisomes is cancers. You can see from these studies that this impairment with peroxisomes can affect its activity or even its overall numbers in the cell. It has recently been reported that cancer patients, especially those with blood or lung malignancies or tumors that have spread through the body, have a higher risk of death or other severe complications from COVID-19 compared to those without cancer. In some cases, cancer patients are 3 times more likely to die from COVID-19. There are other diseases out there that involve defective peroxisomes, and I think it would be very interesting to see if the infection rates or the response to this virus in these individuals is different. It's clear that peroxisomes are important in the antiviral response, but Could defects in the system make a person more vulnerable to infection or to have a worse outcome? More work is clearly needed to prove or disprove this theory. Before I wrap up this section, I just want to give you a summary of what we observed in our day 1 post-infection signature regarding biogroups and pathways. First off, we see a clear induction of inflammatory and immune responses in the lung that involves both innate and adaptive immune type genes. We also note the induction and expression of all 3 types of interferons, alpha, beta, and gamma, and speculated that the previously observed inhibition of type 1 and type 3 interferons with COVID-19 may be happening post-transcriptionally. We also saw genes associated with a bad outcome for infection. This is the JAK-STAT pathway, and observed that a key signaling molecule for this pathway, interleukin-6, was higher in the more severe infection signature compared to the average of all groups. In the genes that went down, pathways associated with the metabolism of lipids and reactive oxygen species were overrepresented. We also saw lots of cell cycle genes decrease with infection and speculated that it might be caused by the viral upregulation of cell cycle inhibitors cyclin D2, P21, and MYC. We also spent some time examining the peroxisome category and speculated that defects in this organelle may be responsible for severe infections to the virus in older individuals, as well as disease states that involve altered peroxisome function. I will deposit a list of the pathway and biogroups that were overrepresented in the 1-day signature of infection in my GitHub account, where you'll coincidentally be able to find the original data that this analysis is based on. As I've said before, the purpose of me doing these videos isn't to show off what I can do, but to gather interest so others take this up and hopefully come up with something that actually helps people. The host signatures to infection we've developed are based on lots and lots of data from many different lab groups and contain insights you could not get with just one study. In our next section, we will further explore our day 1 infection signature by throwing it through the pharmacotab and base-space correlation engine to see what drugs and/or compounds may have an effect on these genes. You will be really surprised at what comes up in our drug screen. Thanks for watching, and I'll see you in the next section. Stay safe.